So first of all, I want to thank the organizers for inviting me to speak at this awesome dengue symposium. And before I start, I actually wanted full disclosure here is in my talk, there's actually only one slide with dengue data from our group, and it's the last slide, right? So I really appreciate being asked here to talk about sort of different approaches, host-target approaches where we're developing broad-spectrum antivirals that won't only be active against flaviviruses, but hopefully other viruses as well. So thanks for letting me tell my story. Okay, this is the problem that other people had. Green button. Here we go. All right. So I'd like to first start out with the scope of the problem, right? So if you go to our DNA sequence databases, there's 202 viruses that have been sequenced that are known to cause disease in man. So how many viral infections or different types of viral infections do you think we can treat with FDA-approved antivirals? The number is 12. That's pretty astounding, right? Hundreds of viruses that can cause disease in man, and we have marketed approved uses for only 12, right? If we add back vaccines, there's overlap, and that's 26 viruses total that we can defend in a pandemic. We know how hard, and by the way, the news gets worse, right? We all here know that hundreds of thousands of viruses that could leak from animals into man. So all of us here appreciate that another COVID's not an if, it's a when. And so what I want to tell you about is, as part of our medical countermeasures sort of portfolio, we really need to start thinking about broad spectrum therapies, right? And so how do we develop broad spectrum therapies? And so I like to think about it as sort of three buckets of targets, right? And so, of course, we can target the virus, and we're starting to do a lot of that, right? Use computational programs, find conserved areas. We certainly have examples of broad-spectrum nucleosides like remdesivir and favipiravir. And probably from a development perspective, these are probably the lowest in risk because we really understand them, right? But I think from the pandemics and how we reacted, the repositioning efforts portends limited award in this bucket, right? So the next bucket I like to call 1 degree of separation from the virus. And so that's sort of a host cell process or structure that directly interacts with multiple viruses types or single virus types. Of course, we can understand entry inhibitors, transport mechanisms that may be common across different types of viruses, or even pH, right? But the development of drug resistance can still occur. And so, of course, the last bucket is the one I'm going to talk about, intrinsic immunity. Right? And intrinsic immunity is a subset of innate immunity. I think about it as the immunity within the infected cell that may or may not require immune effector cells as part of its mechanism. Right? And so this has to do with sort of stress-activated signal transduction, dysregulated metabolism, all cellular processes. We heard about autophagy, right? I mean, these are things that many viruses require. And so the idea is to be able to target them in such a way that we slow down the virus, but we don't affect the viability of the cell. And so for that reason, perhaps they're higher risk with respect to development. But that said, as part of my story today, hopefully you'll start to appreciate how some surprising activities may actually complement how direct-acting antivirals address infection. So I'm going to be talking about sirtuin 2, or SIRT2. Sirtuins are class 3 lysine D acylases, so the most common form of post-translational modification that we know about sirtuins is deacetylation. Right? And so these changes regulate all kinds of cell processes, from epigenetic effects to metabolic effects, and of course viruses require many of these processes. And so there's now an accumulating body of literature that's showing that SIRT2 directed small molecules of very different structural classes are broad spectrum in their effectiveness against multiple intracellular pathogens, including even intracellular bacteria. Importantly, the SIRT2 knockout mouse is healthy and less susceptible to infection. So in this slide, just in pictures, what I just told you in words, right? SIRT2 is an NAD-dependent deacetylase. The chemistry here is just indicating that it can take a substrate protein that has an acetyl group on it, It requires NAD as a cofactor. There's a catalysis where the acetyl group attacks the NAD, nicotinamide is released, the acetyl group attaches to the ADP ribose, that's a product of this reaction, but importantly, the third product, of course, is the product protein absent the acetylation. Right. Now, on the top right here, I'm showing that what's unique about these NAD-dependent deacetylases is that their activity is tuned to the metabolic capacity of the cell, right? And that is because under NAD high conditions, that represents sort of a low capacity or nutrient poor condition in the cell, and of course, many viruses impose that condition on the host cell. All these targets in the bottom corner here, these are all known targets of SIRT2 deacetylase activity, and you can see many different important processes in the cell that are required by different viruses in different ways for their productive infection. And P53 is highlighted because that's one little tiny story that I'm going to tell you about later during the talk. So before I tell you about how these behave as antiviral agents, I do want to talk a little bit about these small molecules. These are not your classic competitive inhibitors. These are allosteric modulators of SIRT2 activity, and a picture speaks 1,000 words. So here we've done some computer modeling, and maybe the middle picture is the easiest, the space-filling one is the easiest to kind of visualize this. So here in this figure, the orange backbone, that's the backbone of NAD, so that's the binding pocket there. The green coming in, that's a lysine, that's a peptide lysine presenting a acetyl group to the NAD. And so that's what this enzyme does. It brings these two molecules together and drives the catalysis. In magenta is one of our compounds, FLS359, and so we've actually shown by enzymatic, by binding studies, and actually the actual co-crystal that indeed this is how these molecules bind. They change the positioning of the two substrates relative to each other, and therefore they're not competitively competing these substrates, but they're changing the kcat of the enzyme. And the last bullet here is really important for me to mention. These modulators are partial inhibitors, so even at maximal inhibition, we never completely shut down SIRT2. Now, we think that's important for the antiviral effects of these compounds because that allows us theoretically to achieve a therapeutic window between inhibiting the virus as well as we can without adversely affecting the physiology of the host cell, and importantly, uninfected cells neighboring uninfected cells. Okay, so big table. So here we have looked at over 20 different virus types, right, across DNA and RNA virus families, unenveloped, enveloped viruses. And if you look at the three middle columns, we're showing the 50% effective concentration for FLS359. This is an early lead in our program. And then EV100, which is a cousin of FLS359 that we're actually sending into the clinic, and we hope to be in people at the end of next year. And then the comparator, right? And in some instances, our compounds are actually more potent than the comparator. But the kicker, of course, is in the second to last column on the right, and that is the comparator, in some cases, is a direct-acting antiviral that is not expected to be effective against any of the other viruses in this table. And then finally, I've summarized some data around other pharmacophores. These are literature compounds that do show activity, again, broad spectrum activity against many of these intracellular pathogens. And if you kind of look halfway down the list, you'll see that Zika and Dengue, particularly tenofin-1, is a SIRT2 small molecule inhibitor that can affect the growth of these viruses. This table summarizes data generated at Everest. We've particularly focused on three viruses: a big, large herpes virus, human cytomegalovirus, hepatitis B virus, and OC43, a beta coronavirus. And what I'm summarizing here is that depending on the virus, we clearly have effects on inhibiting viral transcription, replication, and extracellular genomes. And what I want to kind of stress here is something that's actually quite impressive is we can actually look at the infectivity of the released particles. And here we're showing that we're getting 2 or even 4 log reduction in particle infectivity for coronavirus and for herpes viruses, or for cytomegalovirus. In this slide, I'm showing you data in a viral passage experiment to ask whether do we indeed provide a high barrier to resistance. This is actually in MDCK-infected influenza A cells. And in this passage experiment, we essentially subject the virus to a concentration of drug, the IC99, such that 1% of the virus breaks through. Right, and so you can see in P1 or passage 1, we did quite well here, right? So red is oseltamivir, gray is FLS359, and you see 1% breakthrough in that first passage. So as you sequentially passage these cells, you can see the red bar getting bigger and bigger. That's oseltamivir because that's the evolution of drug resistance in that population of virus. By passage 10, all of the virus is no longer sensitive to oseltamivir. It's very, very tiny. You see that tiny little gray bar in passage 10. That's the FLS359 treated virus, not showing any sign of acquired resistance. So the other sort of profound effect we've observed with our compounds is that we have a very strong effect on cell-to-cell spread. Right, and so in these cultures, we're looking at labeled virus. We start at low MOI infection, like in panel A, and as the infection evolves, you can see some virus actually gets from one cell to neighboring cells by direct cell-to-cell spread. Right. I'm not going to go over these, all these graphs, very complicated dose-response curves, but what we're showing here is that as you increase the multiplicity of infection in this assay, we do not decrease the potency of EV100, right? And that, if you think about it, that makes sense, right? Because we're providing a poor environment for the virus to replicate, and it doesn't matter how much virus is there, right? And so that's the take home there. Like in comparison, ganciclovir clearly is affected by multiplicity of infection. And here I'm coming back to that P53 story, right? And so most of us know that P53 is essentially the cellular switch for apoptosis, and that many viruses, including influenza, we're going back to flu here, actually actively affect the activity of P53. In the case of influenza, early in infection, it actively inhibits the activity of P53 because it essentially wants to keep the cell alive long enough to support productive infection, right? And in the case of SIRT2 acetylation or deacetylation of P53, it turns out that the hyperacetylated state of P53 holds it in an active form that can recognize the cell as being infected, and therefore I need to self-sacrifice myself and eliminate myself by apoptosis. And so with SIRT2 modulation, then we reverse the active inhibition of P53 by influenza, and the cell again dies, right? And so the data is actually shown in the graph. Importantly, the first 2 bars, the red bars, that's tunnel staining, so we're just counting tunnel positive cells in cultures where it's just the cells. Right, so there's no increase in apoptosis with the application of FLS044, which was a precursor of FLS359. However, the green bar, here we're following the MP protein of influenza, and so if you put flu on the cells, you obviously see increased amounts of MP protein. That's brought down by the 044 treatment, but then apoptosis is climbing up because the infected cells are being eliminated by apoptosis and reactivation of P53. So this apoptosis that we observed in MDCK cells, this is actually work from Gary Chan, who's here at SUNY Upstate. Quite frankly, our participation in this collaboration was to give him the drug, but he's showing that SIRT2 can promote the death of human cytomegalovirus positive monocytes, all right? And so we're seeing some of these intrinsic immune things going across cell lines and cell types. And so why is this interesting, right? Well, in the case of CMV, CMV, as you know, can infect many, many different tissues under immunocompromised conditions. And so part of the reason why I can get to all these places is it's thought that the monocytes may actually be the carrier, all right? And so if we can eliminate the positive CMV monocytes, we may, in fact, from a translational point of view, be able to slow down dissemination of the virus systemically, right? And while we didn't look at this in CMV, we did look at this in chikungunya challenge studies. So this is done in mice. This was done in collaboration with Dan Strebolo at OHSU. And I think this experiment kind of also illustrates what is difficult about host targeting, right? I mean, I showed you a table where all kinds of viruses, all kinds of cell types that were able to stop the infection of the virus. But I think for a host cell target, we have to understand, well, systemically, which are going to be the most, the targeted cells or the targeted condition, confluence of conditions that's going to be most affected. And so in this study, we looked at 4 different cousins of FLS 359. And if you look at panels A and B, clinically, there seems to be no difference or no improvement with treatment of our drug. So particularly in panel B, we're looking at foot pad thickness, which is in this model thought to be sort of an outcome of the CHIKV injection in the foot pad causing inflammation. So that's kind of disappointing. If we look at viremia in panel C, 2 days after, in the first peak of viremia, we may have a 2-fold effect on serum viremia, right? But in panels D and E, we're actually showing about tissue viremia. And while chikungunya is not known to be an encephalitic virus, but the virus does travel to the brain in these mouse models, we're showing that we get quite significant reduction of viremia in the brain, both by plaque assay as well as by RT-PCR. And we're pretty sure that this is not an antiviral effect in neurons because our drugs do not cross the blood-brain barrier that well. So part of the challenge is going to be what in the human condition is going to be most subject to this sort of therapeutic window that we can create with these intrinsic immune compounds. And so I promised you a little bit of dengue data. EV100, we're taking into the clinic to treat immunocompromised transplant patients and herpes viruses in general. But we are now starting to probe dengue virus activity. So 359 I mentioned is active, also 673. But it's kind of cool, right? They are also active against these very distinct hemorrhagic fever viruses like Lassa fever and Ebola. And so the vision here is with a more potent molecule, we might be able to address these hemorrhagic conditions that are simultaneously address them for these very different viruses. And so with that, I thank you for your attention. Oh, very important slide, my acknowledgements, right? So, as you can imagine, studying broad spectrum antivirals across all these different virus groups is not an easy task, all right? You've got to set up all these models. And so we are completely dependent on the amazing investigators that we've collaborated with all across the country and at these institutions. So thank you.